All right. So as you all know that we had a major bloom back in 2016 at the Murray River, which invoked considerable management responses, and it was a nightmare for many of the water managers who are present here. But I'm really sorry that I have to bring those memories back. Pretty much, my talk is going to be like, what I presented in 2016 about the toxigeneicity and what we did from there on. So as Nick said, what to do with Victorian sites, but you know that the Murray River bloom, it goes, it's between these New South Wales, Victoria, and South Australian borders. So we tested all the samples from the Victorian part of the Murray River. So that's what I'm going to talk about. Oops, how do you go? Yeah. So basically, these are the samples that we collected. So almost 194 samples were collected throughout this bloom from March 2016 to May 2016. Oops. What is this? Oh. So what we did initially was a multiplex tandem PCR. Basically, we looked at the toxigenicity using PCR-based analysis. So this was a tool developed by our lab back in 2013, where you can actually simultaneously detect 4 different cyanotoxins, that is microcystin, cylindrospermopsin, saxitoxin, and nodularin. So based on this MT-PCR data, what we saw was this bloom had microcystin, saxitoxin, and cylindrospermopsin producing species. No samples were tested positive for nodularins. But what you have to basically see here is, oops, jeez. All right, I'm not going to use this one. Okay, so anyway, what you have to see here is, if you look at the upstream sites, you can see the microcystin population or microcystin producing species were abundant. And then as you move down the river, so you can see when you see after Nea, Piangle, or Weeman, you can see this microcystin boom population starting to rise again. So how do you? Yeah, so if you look in here, you can see this microcystin populations abundant here and as well as here. So which means that this bloom had probably multiple bloom types or multiple toxigenic producing species in there. So we wanted to test that. So basically, if you look in here, so the upstream direction had cylindrospermopsin and microcystin producing species in abundance, and as you move downstream, you can see the microcystin producing species based on this based on the MT-PCR analysis. And we retested many of our samples using conventional PCR, ELISA, and HPLC. And what we could see was these were basically tested with independent primers published from or published in other literatures. And we could detect all these 3 different toxin types in this bloom. And also these was tested, confirmed with ELISA and HPLC. So we detected cylindrospermopsin, microcystin, and saxitoxin was not tested because we didn't see high gene copy numbers for saxitoxin producing species. So we tested only for cylindrospermopsin and microcystin. But what we saw was ELISA and HPLC, it showed a extremely low level production of these toxins, and overall toxins levels were really low and never rising about 1 microgram per liter. Then what we did was we did some additional testing. We looked for anatoxin producing species in the Murray River, because what we know that in Australia we don't see anatoxin producing species. But I used the same samples and I did some nested PCR based approaches, and what we could see was out of the 82 samples that we tested, we could see 59 of them came up positive for anatoxin producing species. So this is the first time that we can confirm that anatoxin producing species are present in Australia, and they're capable, or they have the tool to produce these toxins. So we did some phylogenetic analysis on this, and what we could see was these toxins. So what we could see is in bold, these are the samples that I tested, and these were clustering with Cuspidothrix E. sessionkoi, which is a known anatoxin producing species. A few of those samples were clustering with Anabena or Dolichospermum and Oscillatoria species as well. So we went back and try to see whether if these species they actually produce anatoxin in there. So we tested them with ELISA Abraxas kit. So of the 40 samples we tested, we found that 32 of them came out to be positive for ELISA Abraxas, and 2 samples exceeded the PGV value of 3 microgram per liter, what you see in here. So these were the distribution of anatoxin by ELISA. So what was the microscopy data that we got from Murray River bloom samples? So we know that, or everyone agrees that it was the predominant species was Chrysosporum ovulisporum. So one of the questions that we wanted to ask was like, was this actually correct? And what was the overall microbial population within this bloom? And also we wanted to see those multiple bloom types, basically to like, why did we see those different toxigenicity patterns at one side of the river and on the other side of the river. And we know that there are many limitations. It's a wonderful, wonderful tool, but we know that they've got many limitations. Basically, when we look, what we know that almost half of the cyanobacterial strains in the culture collections have been misidentified. So if we extremely depend on microscopy to ID these sort of species, we may get it wrong. So, and plus, microscopy cannot tell you whether these species can actually produce toxins or not. So we went and looked into the community composition analysis of this bloom by using 16S rRNA sequencing by Illumina MySeq analysis. So this was basically a short-read amplicon sequencing, which means like we are of this 16S rRNA gene is almost 1,500 base pair long. So we amplified almost 300 base pair of that gene, and we did some sequencing analysis using Illumina MySeq. What we saw in this was like, if you look into this first graph of a bar chart here, this is the overall microbial population that we saw in these samples. You could see that a lot of other bacterial population along with this bloom, of which the majority of them, what you see in this blue is cyanobacterial population. And you can see all the other species living or surviving along with cyanobacteria. And also, with this technique, we can also try and find out what could be the microbial population when you don't see a cyanobacteria, especially in the winter season or some of those times. And we can see how these populations actually change when there is cyanobacteria and how they actually interact in the ecosystem. But interestingly, what we saw in the cyanobacterial population, what we see was Dolichospermum as the majority or the major sequence type that we detected. We did not see Chrysosporum ovulisporum in our samples. It was Dolichospermum throughout the samples. So we were actually amused by this result. So we thought, okay, maybe if you look in here, this was basically, we compared our sequence analysis or sequence results with a database. When you get these sequencing results, you compare them along with the database. So here we use Green Genes database. So we wanted to know whether we actually comparing this with the right database. So we went and compared our sequences with another 16S rRNA bacterial database, which is a silver database. Again, in this, what we could see here is that Dolichospermum is the major sequence type that we detected, and we did not see any of the Chrysosporum ovulisporum of all the 194 samples that we tested. So the 16S rRNA summary was like dominant sequence type that we found was like Dolichospermum species. And when we actually blasted them with the NCBI database, what we could see was like we could get many hits like Dolichospermum circinale, Dolichospermum planktonicum. So one of the problems with 16S rRNA gene is that it's very difficult to go down to the species level, and you can get basically go down to the genus level. And Chrysosporum ovulisporum was not detected. And when we actually looked into the diversity or the nucleotide diversity or how identical these species are there based on the 16S rRNA, what we could see here is Chrysosporum ovulisporum and Dolichospermum is only 93.3% identical, whereas if you compare Aphanizomenon with Dolichospermum, it is very much identical, like 99.6% identical. So we were actually not sure whether what we saw in our results was actually Dolichospermum or it could be Aphanizomenon, or Dolichospermum or Aphanizomenon belong to a single genus. So we were not know. But what we know from here is that Chrysosporum ovulisporum is actually different from the Dolichospermum or Aphanizomenon. we went and looked into some of the literatures where people have actually suggested that, Dolichospermum and Aphanizomenon belong to the same genus because their 6' rRNA sequence similarity is higher than 97.5%. That's exactly what we saw in our results as well. So when we looked into the long-read sequencing, so now what we did was, like, we went and looked into another sequencing platform where we used a long-read sequencing technique by amplifying a phycocyanin gene. Phycocyanin gene is a gene which is actually very specific to cyanobacteria. So in our Illumina sequencing, we amplified a 300 base pair gene. So in this case, we amplified a 700 base pair phycocyanin gene, which is going to be much more information rich, and you can actually go differentiate cyanobacteria to its species level as well. So, and we compared all the 3 different species and see how identical they are. So before moving on to a PCR amplification. So what we could see here is that Chrysosporum ovulisporum is 75.7% identical to Dolichospermum, and Planktonicum and Aphanizomenon is almost 88.8% identical. So you can actually clearly differentiate all these different species by amplifying the phycocyanin gene. So we did this using the PacBio circular consensus sequencing method. So this is a heat map, what we saw, what we got from our results. So basically, if you look in here, what you can see is that in our PacBio results, we saw Aphanizomenon as a major sequence type. It was not Chrysosporum ovulisporum, and along with a lot of other cyanobacteria species as well. So then we asked a question like, did our PCR amplification or the primers that we used actually able to pick up Chrysosporum ovulisporum if it was present? So what we did was, like, we took few samples and we spiked our Murray River samples with pure culture of Chrysosporum ovulisporum that we got from Barbara Center way back in 2010. So we isolated the DNA from that pure culture Chrysosporum ovulisporum. We spiked it into our samples, and we amplified using the same PCR primers. We did the same PacBio sequencing. We used the same bioinformatic pipeline. And what we could see here is that if Chrysosporum was present in those Murray River samples, it should be able to pick up, so because we can see it in here. So what that says is that, the primers worked well, our PacBio analysis wasn't wrong, the pipeline wasn't wrong either. So what was it? The Chrysosporum, it was not Chrysosporum, it was Aphanizomenon. So, and then we compared our MT-PCR and the PacBio sequencing data, and what we could see here is that we could see these different multiple bloom patterns. So if you can see in here, so you can see the same different sort of multiple bloom patterns of Dolichospermum and microcystin in different sites. So it was actually correlating well with our analysis. And also what we did was, like, the overall amplicon sequencing correlated well with our toxin type. So what we basically did here is that we took our multiple tandem PCR data, we correlated that with our overall sequencing data we got from PacBio. So what you can see here is the different gene copy numbers, and compare that with different species that we got from PacBio sequencing. So basically what it tells is that the saxitoxin genes most likely came from Cylindrospermopsis. We know that in Australia, Cylindrospermopsis do not produce saxitoxin. It is Dolichospermum circinale. But this is something which we can isolate the species and try and see if this is the actual case. And most of the MCYs came from Microcystis and few unassigned species. So what is it actually? So what could have happened? What we think is that it could be this morphological plasticity is leading us, or confusing our microscopic based sort of identification. And what we saw in the Murray River bloom is actually not Chrysosporum ovulisporum, or it is Aphanizomenon. That's it. And with that, I want to thank my supervisors and all the funding bodies.
So with 46 seconds to go, well done, Nijoy. I guess there'll be a few questions. Do we have any from the floor? Goodness me, no? Michelle.
Thanks for a very interesting talk. Yeah, it's a bit of a dilemma, isn't it? Like, quite a lot of taxonomists have looked at those samples and identified it as a particular species. So it's kind of hard to say that they've got it wrong, isn't it? I mean, there's got to be some kind of reconciliation, I guess, ultimately about the results and the taxonomic stuff, microscopically. Yeah.
Yeah, that's what, because when because I'm not a taxonomist, but when I looked into the literature, what I could see is that the cyanobacterial taxonomy is just like, the taxonomy of cyanobacteria itself is like controversial, complicated, and confusing. Exactly. And so when we look into the molecular studies, what we know is that DNA is basically it's a genetic signature. So I don't know whether if that can go wrong, or that's one of the reasons why we spiked our samples with Chrysosporum ovulosporum pure culture DNA to see if our analysis went wrong. But in that case, it wasn't the case. We could detect Chrysosporum ovulosporum if it was there. So. Yeah, I don't want to, put a lot of confusion or controversy into the taxonomists, but this is what I saw, and I just wanted to present that.
I'm sorry, I was trying to get my head around the taxonomy side. I don't know whether you mentioned it. I was wondering, you mentioned that there was a difference in the toxin profile depending on the place in the river that it was.
Yeah.
You had a cylindrospermopsin cluster, and then you had a microcystin later, further downstream.
Yep.
Did you determine the cause of that in terms of the actual environment in the river, whether it was more turbulent upstream than it was downstream, or...?
We had no, data on those environmental or physiochemical factors that was there during the time of the bloom. So we didn't make any sort of correlation based on that.
Because the heterocystis species will probably persist a little bit more in turbulent systems in areas and microcystis in the more...
It could be, yeah, but we don't have,
areas.
but like I said, I didn't have any data on those sort of, like the pH or the turbidity or temperature. We don't know anything, I don't know anything about that.
Right down the back.
So with the genes you're looking at for the Dolichospermum and the Aphanizomenon, or however you want to pronounce it, have you gone to species of the Aphanizomenon that you're saying it?
Yep, so what you see there is the Aphanizomenon flow sac. So...
Because I'm just thinking so Chrysosporum ovallisporum used to be called Aphanizomenon ovallisporum?
Yes.
Yeah, so you know that you're not using that gene?
So that's what we know, because this gene is not based on, this is a common gene. So the 6-ness RNA gene is present in all bacteria, whereas phycocyanin is present in all cyanobacteria. So if you look into, specifically for phycocyanin gene, so if you look into the identity of these genes, they're actually different. So if you amplify that gene and if you look into the sequence, you can actually differentiate these three from one amongst the other. So what we saw in our result was based on the phycocyanin is Aphanizomenon flows aquae. So that's what our sequencing result came out to be. It was not Chrysosporum ovulosporum. If it is highly identical, like the case of 6-ness where Dolichospermum and Aphanizomenon is 99.6% identical. So in that case, you can say, do they belong to the same genus? But if you look into phycocyanin, it's much more information rich. And that's one of the reasons why we did a long read sequencing, so that we can get more information and we could actually separate them from all three. So based on that, we think it is Aphanizomenon flows aquae.
Any more questions?
So one of the other thing that I wanted to know is, I know that there's a lot of confusion in here, but if anyone has isolated this Chrysosporum ovulosporum from the Murray River sample or the 2016, I would love to have that sample and do more analysis on these to see what exactly is happening there. Thank you.
So thank you very much. It was a great session. I'd just like to make one point, as I was, I guess, a co-author on this paper. Unlikely to solve the fraught world of cyanobacteriology Cyanobacteria taxonomy. But one thing about molecular approaches is that they are highly repeatable. We must not forget that. And I think it would be a good idea if we were using them more as a complementary tool, you know, when we do field work, as an internal control, and also when we deposit samples in culture collections.